Hello, my name is Kristel Bustamante, and today I'm going to talk about specifying the pancreatic islet from biomechanical forces. First, the definition, pancreas, an organ gland that make hormones and secrete enzymes for the digestive process mainly. Pancreatic islets, islets of Langerhans, group of the cells in your pancreas that have several types of the cells, alpha, beta, gamma, and delta cells. Beta cells make the hormone insulin, which helps your body use glucose for energy and helps control your blood glucose levels. Biomechanics, a story of forces acting on and generate with the body and of the effects of these forces of tissues, fluids, or materials used for diagnostic, treatment, or research purpose. Next, the first video.
Welcome to 5 Minute School, and in today's video we're going to be talking about the pancreas. I've included this diagram here, which you can see we have the anatomy of the pancreas. This part is the body, this is the tail, and this is the head. We have the pancreatic duct located here. These are the lobules of the pancreas, and we have the gallbladder, which is located on top. In today's video, I just want to talk about the endocrine function of the pancreas. So it consists of islets of Langerhans, which are also consisting of alpha cells and beta cells. The alpha cells produce glucagon, and the beta cells produce insulin. In today's video, I'm going to be talking about both of these hormones, and videos following on from this will look at other endocrine organs as well. So we have glucagon, which is produced by the alpha cells of the islets of Langerhans. We have the glucagon, which is antagonistic to insulin. It raises plasma glucose concentration. It's stimulated by low plasma glucose levels, and it stimulates glycogenolysis, which is the breaking down of glycogen. It also stimulates gluconeogenesis along with glucocorticoids. Now, it also stimulates lipolysis, the hydrolysis of fat, and ketogenesis, which is the synthesis of ketones by the liver from free fatty acids. Now let's talk about insulin. Insulin regulates plasma glucose concentrations. A rise in plasma glucose levels stimulates the beta cells to secrete insulin. Insulin binds to the target cells and causes intracellular vesicles containing the GLUT4 carrier proteins to translocate to the plasma membrane. And then these carrier proteins promote the facilitated diffusion of glucose into the cells of insulin's target organs. And they are most commonly the skeletal muscles, liver, and adipose tissue. Insulin also indirectly stimulates the activity of the enzyme glycogen synthase in the skeletal muscle and liver, which promotes the conversion of glucose into glycogen. So overall, insulin lowers blood glucose and promotes anabolism.
Okay, next, applications in medicine, biomechanics, a story of how cells sense mechanical forces or deformations and transduce them into biological response. A story of how mechanical forces alter cells, ground, differentiate movement, signal transductions, protein secretions, and transports, gene expression and regulations, determinations, and differentiate subtypes have clear-cut identities. It involves progressive restrictions in their development potentials in which the cells choose a particular fate determinate, but still looks like in its undetermined neighbors. It is applied for the diagnostic and treatment of pancreatic disease like diabetes, which affects beta cells and glucose metabolism. The next video.
Glucose level in the body is regulated by the islets of Langerhans, spherical glands located around pancreatic ducts. They are comprised of beta cells that secrete insulin, alpha cells which secrete glucagon, and several other endocrine cell types. Type 1 diabetes is caused by the loss of the insulin-secreting beta cells. Islet transplantation from cadaveric donors can cure the disease, but donor scarcity requires finding alternative sources. In vitro generation of islets from human embryonic stem cells could serve as an alternative. To enable their production, scientists at the Harvard Stem Cell Institute determined how islets form during embryonic development. In mice, islets start forming around the 13th day of pregnancy. At this time, the core of the developing pancreas is a network of epithelial tubes or cords. The cord cells divide. Inhibition of Notch signaling induces some of the daughter cells to express neurogenin-3, and these cells start differentiating towards an endocrine fate. As the differentiating endocrine progenitors leave the epithelial cord, neurogenin expression decreases and pan-endocrine genes such as chromogranin A are turned on. Cells leaving the cords maintain cell-to-cell contacts. As more cells leave the cord, they push the earlier cells outward and form a peninsula, which will later on form the islet. The first cells that exit the cords turn on glucagon and become alpha cells. Because they were the first cells to bud out, these alpha cells appear at the periphery. About a day later, insulin-producing cells start to appear in increasing numbers. Because these beta cells originate from later progenitors, they are closer to the peninsula base. In the following days, more cells leave the cords and join the peninsulas. Adjacent peninsulas may fuse and the peninsulas grow. As birth approaches, the production rate of new progenitors wanes. Peninsular growth is now driven by division of the differentiated hormonal cells. The initial architecture of the peninsula is maintained in the sphere, and so alpha cells appear as a mantle around a core of beta cells. Peninsulas appear along much of the cords and can be found as a continuous necklace that will later break into individual islets. During the entire process, the forming islets are vascularized. The islets are now ready to sense the amount of glucose in the blood and react by secreting insulin and other hormones into the bloodstream.
Okay. Next, pros and cons. Pros: understanding the biomechanics and learning of each element helpful for studying disease etiology, making decisions about treatment, and evaluating treatment effects. Now, cellular and extracellular microenvironments within pancreatic tissues, understand disease and complications, study for therapies or treatment like replacement or transplantation of beta cells. Cons: expensive and a bit complicated, limited study, not direct diagnostic for treatment.
Therapy for diabetes, we are learning to make beta cells from stem cells for transplantation to patients. Beta cells start to form during the development of the pancreas. In the embryo, the pancreas begins as two buds on the sides of the future intestine. The buds, made of pancreas progenitor cells, grow as the cells multiply. As more progenitors multiply and the buds grow, some of the progenitor cells begin to specialize, moving outwards beyond the final layer of the developing bud. These cells begin to form groups, the beginnings of the islets of Langerhans, the home of the hormone-releasing cells, where mature beta cells will be found. Other progenitor cells polarize, creating holes. The holes begin to merge, forming tubular branches. At the end of these branches, the progenitors begin to form a different type of specialized cell. These acinar cells release enzymes that will flow down the tubes to help digest food. As more progenitors and acinar cells multiply, the tubes elongate, developing the branched tree-like structures found in a fully grown pancreas. As development continues, more progenitors specialize and shed from the branch, forming islets, housing the hormone-releasing cells, including beta cells. Connected to blood vessels, the islets release hormones, insulin to decrease our blood sugar, glucagon to increase sugar levels. Vascular cells found at the tips of the branches release enzymes into the tubes leading to our intestines to help digest food. Understanding how the pancreas forms and how its cells specialize helps in understanding how to make these cells from stem cells in the lab, with a view to one day making beta cells as a therapy for diabetes.
Okay, and the next issues apply in Ecuador. 60 to 80% of the cells in the islet are beta cells, most important cell population to store insulin secretions. Pancreatic islets are a crucial modern system to store ex vivo insulin secretions. Research is continue to refine the understanding of how functional beta cells differentiate. Create a roadmap that could be used to eventually recapitulate either in vitro or in vivo their molecular events that direct progenitors to differentiate into islet endocrine cells. The research. In conclusion, objective identified the determinate or of differentiate of the cell types, in particular the insulin producing beta cells. Studies show biomechanical signaling pathways drive cell fate decision in pancreatic cell type, research beneficial for diagnostic preventions and treatment for diabetes. Now importance of pancreas and its functions. The last video.
Is a mixed gland. A portion of pancreas acts like exocrine gland and another portion acts like an endocrine gland. The cells of endocrine pancreas are called Langerhans, representing 1 to 2% of pancreatic tissue. They secrete two hormones, insulin and glucagon. One, insulin. The hormone is produced by beta cells of Islet or Langerhans. It is a peptide hormone which plays a major role in the regulation of glucose homeostasis. Insulin promotes the conversion of glucose to glycogen in liver and muscle that is stored in these tissues for future use. Insulin is liberated when the blood glucose levels are high, usually when food is digested and absorbed. If sufficient insulin is not produced, glucose level in blood increases and it is excreted through urine. This is called diabetes mellitus. Diabetes is identified by the presence of sugar in urine. Therefore, diabetic patients take insulin injections regularly. Insulin also affects the metabolism of fats and proteins. Unattended diabetes results in wasting of body tissue, reduced eating power, ketone bodies, blurred vision, and kidney problems. They may end up with gangrene and coma. 2. Glucagon is a peptide hormone secreted by alpha cells. Acts on liver cells and stimulates increased blood sugar, hyperglycemia. This hormone stimulates the process of gluconeogenesis, which also contributes to hyperglycemia. Glucagon reduces the cellular glucose uptake and utilization. Thus, glucagon is hyperglycemic hormone.
Okay, thank you.